Hello everyone, my name is Ava Mack and today I'll be talking about progesterone and its many effects on the brain and behavior. Before we begin, I want to talk about why I really wanted to learn more and talk about progesterone. I feel that it often gets overlooked in comparison to estrogen, and it has many more effects on the body beyond its role in regulating menstruation and pregnancy. What makes it so interesting is that its many effects are still not totally understood, and we are constantly learning more about its effects on the body, brain, and behavior. Below, I have a pictorial overview of what I'll be talking about today, and I'll first briefly go over the effects of progesterone on the body, and then I'll discuss its effects that have been found on sexual behavior, mood, and cognition. So first off, some basics about progesterone. It is a steroid hormone synthesized in many parts of the body, but most notably in the corpus luteum of the ovaries, the placenta, and adrenal cortex, as well as Leydig cells of the testes, adipose tissues, neurons, glial cells, and other parts of the body. As you can see below, this is a general synthesis pathway of progesterone, and as a steroid hormone, it starts out as cholesterol, undergoes two reactions, and becomes progesterone. And it exerts its effects on the body through many different mechanisms, but most notably nuclear mechanisms as a transcription factor, and also through intracellular signaling pathways. Now moving on to what progesterone does in the body, I've separated my slides here by sex, but I want to note that throughout this presentation, I will be referring to individuals assigned female at birth as AFAB, and those assigned male at birth as AMAB, just for inclusivity's sake. Also, for the sake of time, I will be focusing more on reproductive organs as is relevant to our course's main topic. So in AFAB individuals, serum concentrations vary throughout the menstrual cycle, being very low during the follicular phase and relatively higher during the luteal phase, as you can see here in this diagram. And during the luteal phase, progesterone promotes follicular growth, and along with estrogen, it thickens the endometrium to prepare the uterus for implantation. And following conception, levels rise after the 8th week of pregnancy and rise to up to 100 to 200 nanogram per milliliter in late pregnancy. Now looking at AMAB individuals, they have very low serum concentrations similar to the follicular phase and also similar to postmenopausal AFAB individuals. And in the AMAB body, it is involved in testosterone biosynthesis in the Leydig cells, spermiogenesis, and also sperm capacitation and the acrosome reaction in the AFAB reproductive tract during fertilization. Now looking at progesterone's role in sexual behavior. In humans, the hormone has largely been suggested to have inhibitory effects. In Marcinkowska et al., daily levels were negatively associated to sexual desire, and AFAB individuals with higher average progesterone levels across the cycle reported a lower frequency of sex. However, in another study, progesterone levels in AFAB individuals were positively associated with greater sexual attraction and greater interest to their primary partner, but not those outside the relationship. These findings have interesting implications from an evolutionary perspective, as it may have been advantageous for females to invest in their mates after conceiving with them, but as we know, modern human sexuality is much more nuanced and much less focused on mating to reproduce, so this requires further investigation. And another demonstration of progesterone's effects on sexual behaviors is with progestins, synthetic progesterone. It has been used as a means of chemical castration and decreases deviant behavior of AMAB sex offenders due to its anti-androgenic properties. Progesterone also appears to have effects on sexual orientation. In one study, prenatal exposure to luteocycline, a synthetic progesterone, was significantly associated with a decreased likelihood of self-identification as heterosexual and increased likelihood of bisexual behaviors. Lesbian and bisexual women have also shown higher overall testosterone and progesterone concentrations than heterosexual women. But a meta-analysis by Harris et al. Indicated mixed findings across the literature, so much more research is needed to understand progesterone's role in human sexual behavior and orientation. Now looking at progesterone's effects on mood, progesterone has been heavily implicated in premenstrual dysphoric disorder, or PMDD. PMDD is a disorder characterized by severe mood symptoms in the luteal phase that are markedly more severe than those seen in premenstrual syndrome. In a study from Coe et al., AFAB individuals with PMDD that had higher or increased levels of progesterone during the luteal phase showed a greater increase in PMDD symptoms relative to healthy controls. Also, allopregnanolone or allo and isoallopregnanolone or iso, which are the progesterone metabolites pictured here, have also been linked to PMDD, and some evidence supporting this is that ratios of iso to allo levels were positively correlated with brain activity and emotion processing areas in individuals with PMDD. And previous studies have suggested that while allo increases PMDD symptoms, ISO antagonizes its effects. So this has made ISO the target as a potential treatment for PMDD, and clinical trials administering ISO, also called sopranolone, to those with PMDD have shown promising effects, which is exciting. Progesterone has also been implicated to affect mood in AMAB individuals, but this is relatively understudied. A study by Losenberger et al. With AMAB participants found that progesterone-mediated modulation of serotonin 1A receptor expression was strongest in the amygdala, orbitofrontal cortex, retrosplenial, and cingulate cortex, as you can see in the PET images shown on the left, all of which are regions which are involved in the symptomology of major depression, and also this receptor, which is inhibitory, is also associated with major depression, and this further highlights progesterone's potential involvement in mood and mood disorders, but really more evidence is needed to fully understand its role, particularly in AMAB individuals. Progesterone has also been shown to affect cognitive domains such as attention, spatial navigation, and memory. In a study comparing the attentional networks of naturally cycling and oral contraceptive using AFAB people, they found that higher levels of progesterone induced an enhanced tonic alertness state or an increased level of vigilance and arousal even when there were no alerting cues. And the authors suggested that this may be beneficial from an evolutionary perspective, as this enhanced alertness could help maintain the safety of a pregnancy while progesterone levels are high. Progesterone may also support cognition in general by influencing cognitive recovery following brain damage. In male rats that had received traumatic brain injuries, progesterone treatment improved their cognitive performance and decreased neurodegeneration compared to brain-injured controls. And the neuroprotective properties of progesterone have also been extended to other animal models of neurodegenerative diseases like Alzheimer's, Parkinson's, or strokes. But the results for clinical trials for human TBI, where they administered progesterone, have had mixed findings, but they do nevertheless present an avenue for further exploration into potential treatment options. In terms of spatial navigation, progesterone may enhance performance. In a spatial navigation task, individuals in the luteal phase had improved performance with the landmark strategy compared to what they had in the follicular phase. And similar to the effects on attention, this could have an evolutionary purpose for enhanced spatial navigation for pregnant individuals. Furthermore, progesterone has been shown to affect spatial memory, as male mice with progesterone receptor knockouts had impaired performance in spatial memory tasks, and progesterone's memory-enhancing effects have also been extended to emotional memory. In a study with AFAB individuals, emotional memory in certain tests were positively correlated with circulating levels of progesterone, and as shown in the graph here below, free recall of emotional stimuli but not neutral stimuli were enhanced in the luteal phase, and the authors suggested that this property could play a role in intrusive memory recall observed in post-traumatic stress disorder, of which AFAB individuals are at a greater risk. So in summation, progesterone appears to have a negative effect on certain sexual behaviors, may influence sexual orientation, may affect mood in both AFAB and AMAB individuals, and may enhance alertness, cognitive recovery, and spatial and emotional memory. But still, there is so much to learn about progesterone, especially in humans and especially in the domains described in this presentation. And the current findings that suggest progesterone's role in mood disorders and neuroprotection have important implications for the treatment of such disorders and conditions. But by understanding and studying progesterone, we can hopefully advance the treatment outcomes for those with these conditions and also just learn more in general about this very fascinating hormone. Here are my reference slides, and I thank you for listening.